Hello guys, welcome to BSc Nursing INC Masterclass. In this class, you are getting the master guidance on university exam preparation. The teaching here is based on Indian Nursing Council revised syllabus, and the teaching is delivered after thorough analysis of both questions and evaluation pattern of many universities of India. So we are continuing our exercise with important essay type questions of applied microbiology for university exam. Along with important essay type question, I am also explaining the possible short, very short and multiple choice questions within that particular essay type question. If you are watching this video for the first time, please refer to the previous videos for more questions and answers. I am explaining questions, answers and writing pattern. So guys, the next important essay type question is define pure culture. Describe different pure culture techniques. So according to the question, we have to give the definition. So the definition will be a pure culture is a culture that contains only one species or strain. Species or strain. For example, Staphylococcus. There are different types of Staphylococcus, but one particular strain is Staphylococcus aureus. So, contains only one species or strain of microorganisms free from contamination by other microorganisms. Pure culture technique refers to the microbiological methods used to isolate, means to separate a single type of microorganism from a mixed population and grow it separately for identification, study and laboratory diagnosis. So now give the broad heading types of pure culture techniques. The first one is tube dilution technique. Principle: a mixed culture is diluted serially in sterile broth or saline so that the number of microorganisms decreases progressively, means gradually. Higher dilution may contain only one type of organism which can then be cultured separately. Draw this small diagram and let us understand. Here in the very first tube, sterile broth or saline is given 9 ml. 1 ml pure culture is given here. Now from this concentration, again 1 ml is taken out to the second tube. Here again 9 ml broth is there and the dilution decreases here. Again from this concentration to the next tube, 1 ml is transferred and 9 ml broth or saline is added. So in this manner, the concentration is gradually that decreases, and that higher dilution, there is a chance that it may contain only one type of microorganism which can then be cultured separately. So the procedure of tube dilution technique, it starts with prepare a series of sterile test tubes containing diluent, transfer a measured amount of sample into the first tube, mix thoroughly and transfer a portion to the next tube that we discussed just now with that diagram. Repeat serial dilution several times. Inoculate diluted samples onto culture media and incubate. Observe isolated growth and subculture to obtain a pure culture. Guys, if you are watching this video on a mobile phone, so then you must watch in a full screen so that you can see the whole content clearly. So once you understand the whole concept, then mute your audio, run again and start writing your notes, and your note will be ready easily. So coming back to the uses of tube dilution technique: isolation of bacteria from mixed cultures, quantitative estimation of bacterial population, quantity is estimated, preparation of inoculum for microbiological studies, and advantages: simple and inexpensive, useful for heavily contaminated specimen. Underline this for MCQ. When the specimen is collected from a patient which is heavily contaminated, in those cases tube dilution technique is done, useful for heavily contaminated specimen. So the next technique is pour plate technique. So the principle: a diluted microbial sample is mixed with molten agar, means agar will be melt and after that it will be used. That molten agar and poured into a sterile petri dish. Individual microorganisms become trapped in the agar and form separate colonies after incubation. Means what? A petri dish will be taken, then the sample will be added containing the pathogen, and then the molten agar will be added. Then it will be incubated. So what will happen? The growth will be observed within and also the surface of the nutrient agar. Procedure: prepare serial dilution of the sample, transfer 1 ml of diluted sample into sterile petri dish, add molten nutrient agar cooled to 45 to 50 degrees centigrade, mix gently and allow agar to solidify. Agar should be solid. Then incubate at appropriate temperature. Observe isolated colonies within and the surface of the agar, because colonies will be found within the agar and at the same time at the surface of the nutrient agar. So now the uses of pour plate technique: isolation of pure bacterial colonies, enumeration means counting of viable bacteria. Viable here is active to cause infections. Advantages: suitable for counting microorganisms, supports growth of both surface and subsurface colonies, because what we studied, the colonies will be formed within that agar and at the surface of the agar. And disadvantages: heat sensitive organisms may be damaged by molten agar, because what we studied, the molten agar will be cooled to the temperature of 45 to 50 degrees centigrade. So in that temperature also many of the microorganisms, they cannot survive. The next technique is sprayed plate technique. Principle: a small volume of diluted sample is sprayed evenly, means equally, on the surface of a solid agar medium. Individual cells grow into separate colonies. Procedure: prepare a serial dilution of the specimen. Place 0.1 ml of diluted sample onto the agar surface. Sprayed evenly using a sterile glass spreader. It's a L-shaped rod. I will explain that. This is the agar surface. This is the L-shaped spreader. Here the sample is placed, and when evenly it will be sprayed, it will look like this. So next is allow inoculum to observe. The sample will be observed, then incubate the plate and observe the isolated colonies. Now the uses of sprayed plate technique. Isolation of aerobic bacteria. Underline this. For MCQ, advantages: no exposure to hot agar like pore plate, easy observation and colony selection. And disadvantages: only surface growing organisms are detected because we are spreading the sample only on the surface of the agar. That's why only surface growing organisms are detected. So guys, the next is streak plate technique. Note it. This is a possible short question of 3 marks. Streak plate technique. Principle: the inoculum is progressively diluted on the agar surface by streaking with a sterile loop, resulting in isolated colonies. Let us understand from this picture. This is called the sterile loop. Inoculum is placed here. Imagine 4 quadrants. Quadrant 1, 2, 3, 4 of a agar surface. These are the agar surface. Now, first the sample will be placed in the first quadrant. This is the sterile loop. Then it will be dragged from this place, quadrant 1 to quadrant 2. Again it will be sterilized, the loop. Again from second quadrant it will be dragged to the third quadrant, sterilized, loop again, then finally to the last fourth quadrant. Now we will describe what we observed in the previous diagram. The procedure: sterile the inoculating loop, take a small amount of culture, streak the first quadrant of the agar plate, flame and cool the loop, drag from the first quadrant into the second, third and fourth. Incubate the plate and observe isolated colonies in the final streaked area. That means in that case the fourth quadrant, the final streaked area. Uses: most common method for obtaining pure culture. Underline this for MCQ. Streak plate technique is the most common method for obtaining pure culture. Advantages: simple, rapid and economical, produces well isolated colonies. So guys, that's it about this question and answer. So if you like the videos, give a thumbs up here, and please drop your queries and feedback in the comment section. If you have not subscribed, please subscribe the channel. So guys, stay tuned for the next video. Till then, take very good care of yourself.